So I want you guys to understand that, as I told you, if we know what the cancer is and how it actually started and what triggers the process which we call as cancer, and how we can harvest the potential and resources present in our immune system to fight against cancer. And if you can recall, I told you that it's not a disease like any other disease, it's an age-related disease. So sooner or later, our body will going to develop, our body will going to lose the control on itself. So that is why sooner or later, the people will going to seek for the solutions. And the most, I must say, viable solution will come from our own immune system, because our immune system has great potential to fight against the bad things. And since the cancer is the bad thing, why not using immune system to fight against cancer? I want you guys to understand and try to think and ponder that what is the difficulty, what are the difficulties faced by the cancer cells? What are the difficulties faced by the immune system to deal with the cancer cells? So that will help you to create your research proposals in the future to address and to fix some potentially important options out of this topic. Okay, take this topic as a challenge for you to create your own research proposals. Here we go. The immune system is supposed to be made up of large number of different types of cells. From these different types of cells, some of them are called as the T cells, some of them called as the beta cells. They cells talk with each other, communicate with each other, and first they get the information, and then they pass on that information to the other members of the immune cells. And whatever you are looking at right now, they are the blood cells. The blood cells, they are made up of different types of immune cells out there. Once the information about the cancer cell is generated, once our immune system know about the bad things, or our immune cells feel something is not nice at this area, the immune cell will started harvesting, will started extracting information about the bad cells. They started talking to the bad cells. They started analyzing the bad cells. They started analyzing that it is different from the normal cells. They are feeling the environment. They are trying to feel the situation. And once they make sure that this is not the right thing, they will start killing the bad cells. The other way is that what we are doing is that we are using our immune system to boost. We are using our immune system to boost the treatment. For example, unfortunately, in some people, if there is a bladder cancer, there is a bladder cancer out there, the most easiest way to deal with the cancer is to undergo surgery. They remove the tumor part. This is the tumor part. This is the part which is made up of cancerous cells. If you can remove these things, this is the most easiest way you can deal with the cancer. But there is no way that you can make sure, there is no way you can make sure that all the cancerous cells are removed from the body. Now, the next thing is that our immune system will always fail to figure it out, the difference between the normal cells and the cancer cells, because the normal cells are eventually converted into cancer cells. And most of the cancer cells, they carry the features which are somehow similar to the normal cells. So there is no way for our immune system to distinguish between these two. How about helping our immune system to know there is a problem there? There is a problem there. For example, the cancer is there. Our immune system has no idea about that cancer. You know what? That is why it is growing. Our immune system missed it. Our immune system believed that this is a normal thing. That is why it is growing. How about sending or putting some pathogen at that place? You can remove the cancer and you can drop some pathogen. You can drop something bad out there, like BCG, like other things, or annotated bacteria at that place. It was a very interesting thought which came into the mind of a scientist. I forget the name of that scientist. Please do search that person who actually put the bacteria within the tumor, who drop the bacteria within the tumor. And what happens? Our immune system is not able to distinguish between the cancer cells and the normal cells, especially at the very early stage. But what about putting the BCG, annotated bacteria or viruses at that place? That will going to attract the immune cells. So when these things are there, that will going to help our immune system to rush to that side. And there will be high chances of our immune system to understand that something is not nice out there. You can increase the immune cells at that place. And that is why they might be able to remove the bad things to that. So this is the most easiest of the approach, but not very efficient, but the logic is there, the theme is there. The other thing is more complicated, is more advanced, is more, I must say, robust. And I want you to understand this thing in particular. मैं इसको मजीद explain करूंगा, just video की वजह से मैं जल्दी बोल रहा हूं, okay? I want to talk slowly on these things. The other thing is that using our immune cells to fix the cancer in our body. Not every immune cell is important. Not every immune cell can be used in cancer immunotherapy. Lots of immune cells are moving from one place to another within our bodies. They are also passing by the tumors as well. वो tumor के पास से भी गुजर के जा रहे हैं. They're also passing by the bad cells, but they fail to understand that this is the tumor. Out of many tumors, out of many immune cells, some of the immune cells are eventually landed in the tumor. They sense this is not nice. They sense this is not bad. We need those immune cells. We need those T cells. We need those immune cells which are able to distinguish between the cancer cells and the normal cells. I can tell you 99.9% of immune cells will fail to distinguish between cancer and the normal cells at the early stage. No way. They're almost similar. But think about the 1, 2 or few cells which are able to distinguish that this tumor is not nice thing. This is weird. This is not like normal cells. We need those T cells. We need those immune cells who are living in the tumor, who are trying to deal with the tumor, who are trying to fight with the tumor. So if you extract the tumor, very important now, if you extract the tumor and then take out the tumor, extract it from the tumor, they are very important immune cells because they understand the cancer, they know the cancer. You grow them, you multiply them, you increase their number, or you can engineer them as well. Actually, the next level is to engineer them and then and after multiplying those immune cells, you can put them back into the person's body. You can put back into the person's body, and that immune cells which you will going to put back into the person's body will eventually search and find the cancer cells and will start fighting against it. It's very effective. It's very logical, and it is very helpful to treat cancer. But this is not the ultimate solution, I can tell you. You can ask me why. This is still not the very ultimate solution. Okay? Another way is that there are certain important instructions which are present on the normal cells which help our immune system to distinguish that this is the normal cell. Those are called as immune checkpoints. Immune checkpoints are the receptors which help our immune cells to feel that this is the normal cell. So when the immune cells connects with the normal cell, those receptors help our immune system to feel this is our own cell. I don't need to fight against it. I don't need to kill it. I don't need to do anything. So just go away. Now think if a similar sort of receptors are present on the cancer cells. Kya hoega? What will happen? The immune cells will come to the cancer cells, try to connect it, and with the help of those receptors, the immune cells will feel this is our own cell, and they go away, and they do not touch the cancer cells. So when the immune cells will be able to feel the cancer cells, they will not going to harm the normal cells, but when they touch the cancer cells, they will not going to harm the cancer cells as well. based on the same principles, because the cancer cells came from the normal cells. They have the same receptors. So it will inactivate the T cell. It will inactivate the immune cell. The immune cell will not going to be activated, will not feel anything special, and that will not going to kill the cancer cells. What if we block? What if we block these connections? What if we make the T cells which cannot be stopped? What if we create some inhibitors to these checkpoints? The inhibitors will not allow the proper connection between the T cells and the cancer cells, and that is why the immune cells will not going to be deactivated. They will stay and they will get activated and start doing their job. And what is their job? Their job is to start killing the cells. So this is the thing that through which we able to fight against, we can use our own immune system to fight against the cancer. I will elaborate these things more. If you have any question, you can speak right